So the topic that we are going to learn today is fatty acid oxidation. But before moving on to that, let's learn about the stages of fasting. So the first 4 hours, or the 1 to 4 hours, the body is in a well-fed state and there is an abundance of glucose, so glycolysis takes place and the body gets its necessary ATP. The second stage is 4 to 6 hours after food, that is the early fasting stage. In this stage, the liver glycogenolysis takes place, and hence we get glucose which enters glycolysis, and thereby ATP, or the required ATP for the body, is produced. And the third stage is 16 to 48 hours after food, which is commonly known as the fasting state. And it is in this fasting state where there is a deficiency of glucose and gluconeogenesis has to take place. Gluconeogenesis means making glucose from non-carb sources. So in order for gluconeogenesis to take place, beta oxidation of fatty acid must take place to provide ATP, because gluconeogenesis requires ATP, and for that ATP, beta oxidation of fatty acid takes place. And 2 to 5 days after food is what we all know as starvation, and during this period or this time, the process that takes place is again fatty acid oxidation or beta oxidation along with ketone body synthesis. So from this, what we should understand is that the oxidation of fatty acid, be it beta oxidation or any other type of oxidation, takes place in the fasting state as well as in the starvation state. The fasting state and the starvation state. So as we all know, there are different types of fatty acids. There are saturated, unsaturated, and many other types of fatty acids around us. So for different types of fatty acids, different modes or types of oxidative methods are required. So these are the different types of fatty acid oxidative methods. The first one is beta oxidation. It is done for saturated fatty acids. Secondly comes the modified beta oxidation, which is usually done for unsaturated fatty acids. Thirdly comes the minor oxidative pathways like alpha oxidation and gamma oxidation. Last but not the least comes the oxidative pathway for odd chain fatty acids. So many of you might be wondering, what is a fatty acid or what is its structure? In fact, it is crucial to understand its structure in order to understand the processes of oxidation. So a fatty acid basically consists of an acyl group attached to a carboxylic acid group. So what is an acyl group? An acyl group is nothing but a carbon chain, irrespective of the number of carbons in it. It can either have 2, 3, 4, any number of carbons in it. So an acyl group with 2 carbons is known as an acetyl group. An acyl group with 3 carbons is known as a propionyl group, and an acyl group with 4 carbons is known as a butyryl group. So in short, it can be depicted as below, with the part that I'm drawing as the acyl group. See, this is the acyl group, and the acid group is also depicted here, with the carbon nearest to the acid group being the alpha carbon and the second nearest being the beta carbon. Now moving on to the most important topic of discussion, that is beta oxidation, which is frequently asked as a question. So what is its definition? Well, it is defined as the successive cleavage or oxidation at the beta position of the fatty acid, which often results in the release of a 2-carbon group known as acetyl-CoA. So this acetyl-CoA then enters the TCA cycle, which ultimately results in the production of ATP. That is the whole mechanism behind this. So looking at the diagram drawn below, you can see that the cleavage takes place at the beta position of the fatty acid, which ultimately results in the production of a 2-carbon group known as acetyl-CoA and the remaining part of the fatty acid group, which is often 2-carbon less. And this process is successively repeated and more acetyl-CoA is produced. Now where does beta oxidation take place? Beta oxidation takes place in the mitochondria, especially the mitochondria of liver, muscles, as well as adipose tissue. Now that we have covered what beta oxidation is and the sites of beta oxidation, the steps must come about pretty easily. So we know that the site of beta oxidation is mitochondria. So the fatty acid must be transported into the mitochondria. In order for the fatty acid to be transported into the mitochondria, it first has to be activated. So the first step is the activation of fatty acid. The second step is the transportation of this activated fatty acid to the mitochondria, and the third step is beta oxidation proper. Now let's take a closer look or a detailed look at each of these individual steps. So the first step, as I mentioned earlier, is the activation of fatty acids. The activation of fatty acids is nothing but the conversion of fatty acid, which as I mentioned earlier is an acyl group attached to an acid group into an acyl-CoA with the help of thiokinase enzyme or acyl-CoA synthetase enzyme. And when you hear the name synthetase, you must remember that ATP is used. In this case, ATP is converted into AMP, wherein two high energy phosphate bonds are being used. It is the only energy requiring step of beta oxidation. After all, beta oxidation is used to produce energy. However, this is the only step that requires energy, or ATP, and this takes place in the cytoplasm. And thiokinase or acyl-CoA synthetase is an enzyme that is present in the outer mitochondrial membrane. In this diagram, you can see the outer mitochondrial membrane containing thiokinase enzyme or acyl-CoA synthetase enzyme. You can see the fatty acid in the cytoplasm is getting converted into acyl-CoA in the cytoplasm itself, and here you can see that ATP is converted into AMP and two high energy phosphate bonds are used. So the enzyme is present in the outer mitochondrial membrane and the entire step takes place in the cytoplasm. So after the completion of the first step of beta oxidation, that is the activation of the fatty acid, this activated fatty acid must be transported into the mitochondria via the mitochondrial membranes. So this is achieved by a transporter known as carnitine. Carnitine is beta hydroxy gamma trimethyl ammonium butyrate. Moreover, the synthesis of carnitine transporter requires a large amount of lysine and methionine in the form of S-adenosyl methionine. Now the carnitine transporter is composed of the following enzymes mainly. So they are composed of the CAT enzymes, CAT1 and CAT2. The full form of CAT is nothing but carnitine acyl transferase. So there is carnitine acyl transferase 1 and carnitine acyl transferase 2, which is known as CAT2. The third enzyme involved in carnitine transporter is translocase enzyme. Now I am going to diagrammatically represent the second step of beta oxidation, that is the transport of the activated fatty acid into the mitochondria. Now further elaborating on the diagram that we have drawn earlier, we can see that the activated fatty acid or acyl-CoA can freely pass through the outer mitochondrial membrane and react with the carnitine residues present in the intermembrane space. So acyl-CoA reacts with carnitine in the presence of CAT1 enzyme in the intermembrane space to form acyl-carnitine and CoA. This acyl-carnitine then passes through the inner mitochondrial membrane into the mitochondria with the help of translocase enzyme. Now in the mitochondria, the acyl-carnitine then reacts with CoA. Acyl-carnitine then reacts with coenzyme A in the presence of CAT2 enzyme, where CoA again replaces carnitine to form acyl-CoA, and this carnitine re-enters the intermembranous space to replenish the carnitine residues, whereas the acyl-CoA formed enters beta oxidation proper. Now let us refresh all that we have learned till now. So I'm marking the cytoplasm here, and this is where the first step of, or the first preparatory step of beta oxidation takes place, that is the activation of fatty acid into acyl-CoA. The activated acyl-CoA then passively passes through the outer mitochondrial membrane into the intermembrane space, and in the intermembrane space, with the help of the carnitine transporter, the acyl-CoA then reaches the inside of the mitochondria. Now the activated fatty acid or acyl-CoA has reached the inside of the mitochondria. Now the only step remaining is the third step, that is the most important step, that is beta oxidation proper. Now let's move on to the steps of beta oxidation proper. As we have discussed earlier, we now know that the acyl-CoA has reached inside of the mitochondria. Now let us represent the alpha and beta carbons of the acyl-CoA. As we know that from the definition of beta oxidation, we know that here the cleavage or oxidation takes place at the beta position of the acyl-CoA. So at first, a dehydrogenase enzyme acts on the acyl-CoA. Since it acts on acyl-CoA, it is known as acyl-CoA dehydrogenase. So this leads to a formation of a double bond between the alpha and beta carbon. Hence, it is known as enoyl-CoA. The compound found is known as enoyl-CoA. And on enoyl-CoA, upon the addition of water and action of hydratase enzyme, what happens? A hydroxyl group or an OH group is added to the beta carbon. Hence it is known as hydroxyacyl-CoA. And on hydroxyacyl-CoA, what happens is again a dehydrogenase enzyme acts. and since it acts on a hydroxyacyl CoA, we call it as hydroxyacyl CoA dehydrogenase. This results in the removal of hydrogen and the formation of a ketone group at the beta position, so it is known as ketoacyl CoA. And lastly, on the ketoacyl CoA, the thiolase enzyme acts, which results in the formation of an acyl CoA group that is 2 carbons less when compared to the original compound. And why is it 2 carbons less? Because a 2 carbon compound, 2 carbon unit known as acetyl CoA is released, and this acetyl CoA ultimately enters the TCA cycle. So remembering the names of the enzymes is quite easy. At first, a dehydrogenase enzyme acts, that results in the formation of a double bond, that is enoyl CoA, and then the hydratase enzyme acts, which results in the hydroxylation, that is hydroxyacyl CoA, and then again a dehydrogenase enzyme acts, which results in dehydrogenation, which ultimately leads to the formation of a ketone group or ketoacyl CoA. So the order is dehydrogenase, hydratase, and again dehydrogenase. And finally, on ketoacyl CoA, the thiolase enzyme acts. The thiolase enzyme acts, which ultimately results in the release of the 2 carbon unit known as acetyl CoA, which leaves an acyl CoA that is less in 2 number of carbons when compared to the initial compound or the initial acyl CoA compound. And one thing that we must not forget is, in all the dehydrogenase enzyme reaction steps, an energy compound is formed. In case of acyl CoA dehydrogenase, FADH2 is formed, and in case of hydroxyacyl CoA dehydrogenase, NADH2 is formed. FADH2, as we know, translates to about 1.5 ATP, whereas NADH2 translates to about 2.5 ATP, and the acetyl CoA released, which enters the TCA cycle, translates to 10 ATP. Now the newly formed acyl CoA compound, which is 2 carbon atoms less, again enters the beta oxidation cycle, and this process or this cycle is repeated till the entire acyl CoA compound is broken down into 2 carbon units known as acetyl CoA. Now the next topic that we are going to discuss is regarding the energetics of beta oxidation cycle. Now in order to find the energetics of the beta oxidation cycle, one must know the amount or the number of FADH2 produced, NADH2 produced, and obviously the end product, that is the acetyl CoA produced, because it ultimately enters the TCA cycle that corresponds to 10 ATP. So as we discussed earlier, in order to find out the energetics of beta oxidation cycle, one must know the number of NADH2 and FADH2 produced, and also the number of acetyl CoA produced. In order to find out the number of NADH2 and FADH2, one must know the number of beta oxidation cycles that has taken place. Beta oxidation cycles. So this can be found out by a simple formula known as N by 2 minus 1, where N is the number of carbon atoms in acyl CoA, and the number of acetyl CoA can be found out by the formula N by 2, where N is again the number of carbon atoms in the acyl CoA. So with that, let's move on to an example. Let's take the case of palmitic acid. Let's take the case of palmitic acid, which is a 16 carbon compound. So this is the fatty acid that most commonly undergoes beta oxidation, and it has 16 carbon atoms. So we can find the number of NADH2 and FADH2 via the formula N by 2 minus 1. So N by 2 minus 1 in this case is 16 by 2 minus 1, which is equal to 8 minus 1, that is 7. So 7 cycles of beta oxidation takes place. So in 7 cycles of beta oxidation, we get 7 NADH2 and 7 FADH2. So 1 NADH2 corresponds to 1.5 ATP, 2.5 ATP, whereas an FADH2 constitutes about 1.5 ATP. So from that we get that around 28 ATP is produced in total, and the number of acetyl-CoA. Number of acetyl-CoA can be found out by the formula N by 2. In this case it is 16 by 2, which is 8, and 8 acetyl-CoA is formed. We know that 1 acetyl-CoA enters the TCA cycle to produce 10 ATP. So 8 acetyl-CoA results in 80 ATP. So adding them both, we get a total of 108 ATP. Earlier we had studied that the first step of beta oxidation, or the preparatory step of beta oxidation, included the activation of fatty acid into acyl-CoA, and this step required the presence of thiokinase enzyme and ATP, which was converted into AMP. So as mentioned here, 2 high energy phosphate bonds are used. Now since these 2 high energy phosphate bonds are consumed or used, we have to deduct that from the total amount. So the net ATP produced in the oxidation of 1 palmitic acid is 106 ATP. So now with that we have come to the end of this topic.